Biology, the basis of life. The study of physical structure, chemical processes, molecular interactions, physiological mechanisms, development, and evolution surrounding all living organisms. DNA is what makes us who we are, the blueprint of every individual on Earth. It is made from molecules called nucleotides, and the order of them determines our genetic coding. Genetic coding is the set of rules by which information encoded in DNA is translated into proteins by living cells. While our genes determine the appearance of us, our height, eye color, and many body features, genes also code our behaviors, health, and cognitive thinking. Now, when you're writing a paper online, it's easy to go back with autocorrect or the search bar for mistakes. You can make infinite changes to perfect your outcome. Unfortunately, that's not how our genetic coding has worked until now. CRISPR-Cas9, standing for Clustered Regularly Interspaced Short Palindromic Repeat, is a new technology that allows for gene editing. It guides molecular scissors to a target section of DNA that can disable or repair a gene, inserting the correct gene in place. Initially discovered in 1987 but not used as a safeguard against bacteriophages until 2007. CRISPR is still a very new technology, but it is currently being used as a diagnostic tool for detecting infectious and non-infectious diseases, such using a person's saliva, blood, or urine. It detects and destroys DNA from bacteriophages during subsequent infections. The most recent area of medicine that CRISPR is expected to be used in is to evaluate genes targeted by chemotherapeutic drugs and identify new patterns to reduce or eliminate resistance to chemotherapy and lung cancer. This can and will drastically impact the world of medicine by being able to detect illnesses early on that there is no current treatment for. CRISPR will be able to essentially take these diseases out of our genetic coding. CRISPR is expected to have a never-before-seen impact on the world, affecting it medically, environmentally, and agriculturally. Medically, it will be able to treat Alzheimer's disease because CRISPR-based platforms have been developed to identify the genes controlling the cellular processes that lead to neurodegenerative diseases like Alzheimer's and Parkinson's, hopefully leading to new treatments. It will also treat HIV. CRISPR has been successful in removing the virus's DNA from the patient's genome. Other genetic sequences will likely be found that eliminate HIV, herpes, hepatitis, and other dangerous viruses. Lastly, CRISPR will be able to develop new drugs. Pharmaceutical companies such as Bayer are investing hundreds of millions of dollars to develop CRISPR-based drugs to treat heart disease, blood disorders, and blindness. Environmentally, it will eventually remove malaria from mosquitoes. Scientists have created mosquitoes that are resistant to malaria by deleting a segment of mosquito DNA. The ultra mosquitoes pass on the resistant genes to 99% of their offspring, even when they mated with normal mosquitoes. It will also improve livestock. CRISPR has been utilized in China to delete genes in livestock that inhibit muscle and hair growth to grow a larger stock for the country's commercial meat and wool industries. This could become a common way in the future to expand livestock industries. CRISPR will also reduce our need for plastic. It can be used to manipulate a type of yeast that transforms sugars into hydrocarbons, which can be used to make plastic, greatly reducing the need to rely on petroleum-based resources for plastics and easing the stress off the environment. Agriculturally, researchers are using CRISPR to discover new ways to improve crop disease resistant and environmental stress tolerance in plants. If successful, this could result in new crops to help feed the global population. CRISPR proved effective on October 28, 2016, when the first human injection was carried out at the West China Hospital, involving a patient with aggressive lung cancer. The researchers extracted the patient's immune cells from a blood sample and then used CRISPR editing to disable a gene in them. That gene was turned off codes for a protein called PD-1, which usually slows down a cell's immune response, allowing cancer to grow out of control. These PD-1-free immune cells were then cultured in the lab and injected back into the patient. The aim is that they'll now proliferate in the patient's body and attack and destroy cancerous cells. Lead researcher Li Yu stated that the initial treatment went well. It is predicted that this biomedical advancement will trigger a Sputnik 2.0, a biomedical duel on progress between China and the United States, which is best for both of us, as competition usually improves the end product. I hope to one day be able to work with CRISPR technology, specifically working towards a cure for inflammation of the joints. It is medical advancements like this one that continue to inspire me to pursue a degree in biology and become a doctor. This is Isla Herrender from Johns Creek, Georgia, and I want to thank you for watching.